When you hear the phrase fungal infection, I bet the first image that pops into your head is just some sort of cosmetic annoyance.
Right? Like a stubborn case of athlete's foot.
Exactly. Or maybe a localized toenail problem. And the assumed solution for that is a quick trip to the pharmacy for, an over-the-counter topical cream.
Yep, you just apply it for a few weeks and move on.
Right. You assume the keratin clears up and that's the end of the story.
I mean, that's the popular misconception. But the truth is, those OTC creams don't even penetrate the thick keratin of a nail plate anyway.
Wait, really?
Oh, yeah. To actually cure something like that, you're looking at months of oral drugs, like terbinafine. But more importantly, and this is crucial, the actual focus of modern antifungal pharmacology isn't about curing a rash.
It's operating on a much darker plane, isn't it?
Absolutely. Since the 1980s, the entire field has been focused on invasive, life-threatening systemic mycoses. We are talking about pathogens that invade the bloodstream.
Things that cause massive fungal balls in the lungs.
Yes. Or fungi that cross the blood-brain barrier. In a highly immunocompromised patient, these organisms can literally cause systemic collapse within a matter of days.
The stakes are just incredibly high. And it immediately highlights this massive pharmacological disparity that we're going to get into today. Okay, let's unpack this. We have dozens of classes of antibacterial drugs, right?
Oh, absolutely. Beta-lactams, macrolides, fluoroquinolones.
Tetracyclines, the list goes on and on. Yet, decades into modern antimicrobial research, we rely on roughly 4 major classes of systemic antifungal drugs. Just 4.
And, it's really not for lack of trying.
So our mission for this deep dive into the source material is to figure out why. We're going to unpack this surprisingly small arsenal of systemic drugs, the fascinating history of how we found them, and why fungal infections are rapidly becoming a global health priority.
It really all comes down to a brutal biological reality, the core problem of what we call selective toxicity.
Right, because bacteria are prokaryotes, which gives drug developers all sorts of unique targets. But
fungi are eukaryotes, just like human cells.
Which is the terrifying part.
It is. Because they share our ribosomal structures, they have a similar nucleus architecture, and they use the same major metabolic pathways. So we are left with an incredibly narrow therapeutic index.
I was thinking about this biological overlap, and it's basically like trying to selectively poison a highly invasive weed in a botanical garden.
That's a great way to picture it.
Right. But the weed and your prize-winning orchids share the exact same cellular biology. If you deploy a chemical to melt the weed's roots, the orchids just dissolve right alongside it.
Exactly. The risk of friendly fire, of profound toxicity to the human host is huge. So the entire history of this field has been this desperate hunt for minor structural variances.
Like trying to find a subtle typo in the fungal blueprint.
Right. And the first major typo we actually managed to exploit involves the cell membrane. Mammalian cell membranes rely heavily on cholesterol.
It maintains their structural integrity and fluidity.
Right, yep.
But fungi use a sterol that is functionally identical, but structurally just a little bit distinct. It's called ergosterol.
Ergosterol?
Yeah. Because of a slight difference in its chemical side chain, it provided the very first viable target for systemic treatment.
And to target it, scientists literally had to dig through the dirt.
He really did.
The origin story here is one of the most fascinating pieces of 20th century science. So we have Elizabeth Hazen, a microbiologist at the New York State Department of Health back in the 1940s, and Rachel Brown, a chemist.
Right, in Albany.
Yeah. And they were essentially running this massive grassroots mail-order drug discovery program. Hazen would manually screen hundreds of soil samples, often sent in by friends and culture whatever she could find.
She was looking for actinomycetes, specifically.
Exactly. She'd test them for antifungal activity and then mail the promising cultures in mason jars to Brown, who would isolate the active chemical compounds. It was a total brute force screening process, but it paid off. From a soil sample collected on a dairy farm in Virginia, owned by Hazen's friends, the nurses, they isolated a soil bacterium called Streptomyces norsei.
And by 1950, they had extracted a compound that was highly active against fungi. They named it nystatin, in honor of the New York State Health Department.
Which is a great breakthrough, but nystatin was way too toxic for systemic intravenous use.
Right. You can't just put it in an IV. It's fantastic for oral thrush or topical stuff. But they needed something that could circulate in the blood to fight deep tissue infections.
So what did they do? They just kept screening dirt.
They finally struck gold with a soil sample from the Orinoco River in Venezuela. They isolated a different bacterium, which yielded amphotericin B, in 1955.
And for almost half a century, that was the undisputed heavyweight champion of antifungals, the broad-spectrum gold standard.
But how does it actually work?
Well, these drugs are called polyenes. And amphotericin B is amphipathic. It has a hydrophilic side and a lipophilic side. It inserts itself directly into the fungal cell membrane, binding to that ergosterol we talked about.
And then it just punches holes in it, right?
Literally.
Yep.
Multiple molecules aggregate and form a physical pore in the membrane. Vital ions like potassium just leak out, and the fungal cell dies.
But here's the catch with that weed and orchid shared biology. Amphotericin B's affinity for ergosterol is high, but it's not absolute. It binds to human cholesterol, too.
It does. The collateral damage is severe. It earned the drug an infamous clinical nickname in hospitals.
Amphoterol.
Amphoterrible, exactly, because when it binds to cholesterol, it punches holes in human cells. The kidneys take the biggest hit. Nephrotoxicity is a near-universal complication.
Plus the infusion reactions.
Oh, the shake and fever reaction. Patients get violent rigors, high fevers, a massive inflammatory cascade.
It sounds awful. But we did eventually manage to make it safer, right? Not by changing the drug, but how we deliver it.
Yes, through liposomal wrappings. We created a formulation known as ambisome. You basically wrap the toxic amphotericin molecule inside a lipid bilayer.
Like a chemical decoy.
Precisely. It keeps the drug sequestered away from the human kidneys, but when it hits the fungal cell, it releases the drug right at the site of infection. It salvaged amphotericin B as a viable clinical tool for really severe infections like mucormycosis.
But still, relying on a drug that violently rips holes in cell membranes is inherently perilous. So, since physically punching holes causes so much collateral damage, researchers had to figure out how to stop the fungus from building the membrane in the first place.
Right, which takes us out of the soil samples and into the realm of synthetic industrial chemistry. This is the azole class.
Things like fluconazole.
Exactly. Instead of attacking the mature membrane, azoles are metabolic saboteurs. They target an enzyme called CYP51.
And without that enzyme, the fungus can't manufacture ergosterol.
Right, the fungal membrane gets flooded with defective precursors, and cell growth just halts.
Okay, here's where it gets really interesting. If they are just starving the cell of ergosterol, why do azoles cause so many crazy drug interactions, like with statins or warfarin?
That's the catch.
The answer is in the target itself. CYP51 belongs to the cytochrome P450 superfamily.
Which humans have too.
Tons of them. It's an ancient, highly conserved family of enzymes. Humans use our P450 enzymes, especially in the liver, for metabolizing drugs. In fact, a human enzyme called CYP3A4 metabolizes roughly 50% of all prescribed medications.
Wow. So the azoles aren't perfectly selective.
Not at all. They inhibit our liver enzymes too.
So if a patient is on a statin for cholesterol, and you give them an azole antifungal,
The patient's liver abruptly stops clearing the statin. The drug concentration in the blood skyrockets, which can cause severe muscle breakdown or internal bleeding if it's a blood thinner like warfarin.
which means managing these infections is just an incredibly dangerous balancing act. It requires constant monitoring, which is why drug developers kept looking for the holy grail of selective toxicity, a target in the fungus that human biology completely lacks.
And they found it, right? The fungal cell wall.
Yes, humans only have a cell membrane. We have no cell wall whatsoever. But fungi use a polymer called beta-1-sorry-3 glucan to build this rigid outer wall.
And this led to the echinocandins, like caspofungin.
Exactly. Derived from another fungus found near the Lozoya River in Spain. Because human cells don't have a cell wall, this class of drugs, which inhibits the enzyme that builds that wall, is incredibly safe.
It's the safest target we have. No kidney damage like amphotericin, no liver enzyme issues like the azoles.
But pharmacology is always a trade-off.
Right, there's always a catch.
The catch here is their massive molecular size. They have zero oral bioavailability. You have to administer them via. And worse, they can't cross the blood-brain barrier.
So if a patient has a fungal infection in their central nervous system, like cryptococcal meningitis,
The safest drugs we have are practically useless.
which forces us to look at a fourth major target, nucleic acid synthesis. And this involves a drug called flucytosine.
Yeah, flucytosine, or 5-FC. This one operates through sheer molecular deception.
It's basically a Trojan horse.
That is exactly what it is. To the fungus, it looks like a standard building block for DNA. The fungal cell actively imports it.
And then what happens?
Well, the fungus has an enzyme called cytosine deaminase, which converts that harmless-looking drug into a brutally toxic compound called 5-FU.
And human cells barely have that enzyme, so we don't get poisoned.
Right. But, and this is a big but, you almost never use flucytosine alone. Fungi mutate incredibly fast. If you use it alone, the fungus just mutates its transport enzymes and becomes resistant in days.
So you have to use it with something else.
It's all about synergy. You pair it with amphotericin B.
Ah, so the amphotericin punches physical holes in the membrane.
Exactly, which completely bypasses the need for the fungus to import the Trojan horse itself. The holes just artificially flood the cell with flucytosine.
That is amazing. But, despite all these brilliant pharmacological tricks, we've seen some terrifying systemic failures. Which brings us to the harsh reality of how these pathogens are behaving in the real world.
It really shatters the idea that fungal disease is just something confined to hospital cancer wards.
Like the 2012 fungal meningitis outbreak here in the U.S.
A completely sobering event.
A compounding pharmacy in Massachusetts shipped out thousands of vials of steroid injections, just standard shots for back pain. But their sterile process was broken, and the vials were contaminated with a mold called Exerohilum rostratum.
People were walking into normal outpatient clinics for a routine back injection and inadvertently getting live mold spores injected directly into their spinal fluid.
And because it was in the central nervous system, the safest drugs, the echinocandins, couldn't reach it.
Doctors were forced to use massive doses of azoles and amphotericin for months. The adverse effects from the therapy were agonizing. Dozens of people died.
It really proved that a single breakdown in quality control can cause a lethal outbreak in totally healthy people. But the fungi are also just proactively evolving against us, which is the resistance crisis.
This is what keeps infectious disease specialists up at night. Candida auris is the prime example right now.
First found in patients here in Japan in 2009, right?
Yes, and now it is a globally spreading, multi-drug-resistant pathogen. It sits on the World Health Organization's critical priority tier. We are seeing strains that are resistant to all three major classes of drugs.
But the way they're developing this resistance, this is a genuinely contested debate in the field, right?
Oh, absolutely. What's fascinating here is when we look at azole-resistant Aspergillus fumigatus, historically we blame drug resistance on doctors overprescribing pills to patients.
Right, just like with bacterial antibiotics.
Exactly. But researchers started sequencing the genetics of this resistant Aspergillus, and they found these highly specific resistance mutations.
But not just in hospital patients.
Right. They found the exact same mutations in commercial compost, in soil samples, and on agricultural flower bulbs.
Wait, flower bulbs, like tulips?
Yes, the mutations match the strains killing patients in the ICU patients who had never taken an antifungal drug in their lives.
So the resistance isn't starting in the hospital.
It connects to the bigger picture of applied ecology. The agricultural industry sprays immense volumes of azole fungicides globally to prevent crop rot.
So they are likely selecting for resistant strains in the environment before a patient ever breathes them in. We're basically running a global pharmacology experiment in our farm soil.
It is a massive selective pressure, and it really pits global food security directly against the survival of vulnerable medical patients.
So what does this all mean? We have these massive environmental pressures, an incredibly small drug arsenal, and extremely vulnerable patients.
It points to a grim paradox in modern medicine. Think about our greatest medical miracles today: organ transplants, advanced cancer chemotherapy, intensive care units.
They all save lives by suppressing the human immune system.
Exactly, which ironically expands the exact population that is most vulnerable to opportunistic fungal infections. A fully functioning immune system is really the main reason we aren't constantly consumed by the fungi all around us.
And we also have one other major natural defense, right? The thermal barrier.
Yes, our body temperature. Most environmental fungi thrive at ambient, outside temperatures. They simply cannot survive at a human's core body temperature of 98.6 degrees Fahrenheit or 37 degrees Celsius.
It's an ancient evolutionary shield. But this brings up a terrifying climate angle. There's a prominent mycologist, Arturo Casadevall.
Right, his hypothesis is chilling. He suggests that as global baseline temperatures rise due to climate change, environmental fungi are experiencing constant pressure to adapt to hotter conditions.
So if a mold adapts to survive at 35 degrees Celsius out in the wild,
the evolutionary leap to surviving at 37 degrees inside a human lung becomes trivial. This is actually highly suspected to be the trigger behind the sudden global emergence of Candida auris back in 2009.
it really leaves you with a chilling thought to ponder. We've spent this entire deep dive looking at the toxic, desperate chemistry we use to fight these eukaryotic cousins of ours. But our primary defense has never been the drugs. It's always been our body heat. So if the outside world warms up to match our internal body temperature, will our ancient thermal field against fungi simply melt away?